Hello everyone. I will talk about recent work on the state and trait dynamics of the human brain measured using resting state fMRI in general population in relation to behavior. Spontaneous fluctuations of brain activity at resting state are embedded in time and space, exhibiting rich spatial-temporal information that varies within subject, which we call state variance, and between subject, which we call trait variance. Still, the joint properties of state trait fMRI signal variation remains poorly understood. An individual's mental state at any given time may be influenced by many factors that directly affect the circuit activity underlying behavior. Or there might be other dimensions that contribute to variability, such as time-varying signal dynamics. To this end, our question is if there is a reproducible neural feature set that reflects both state and trait brain dynamics and relates to individual phenotypes across multiple behavioral domains. The analysis of moment-to-moment changes in co-activation patterns, or CAPs, assumes a single brain state per each fMRI time frame. Using 4 15-minute run of resting state fMRI data from 337 unrelated general population in the HCP dataset, we identified CAPs recurring over time across subjects. Here, we split the subjects into two equal-sized groups to create a parcel by concatenating time series data metrics for each split, and apply the CAP analysis separately, which were then repeated over 1,000 permutations. The time frames are clustered based on spatial similarity using k-mean clustering, and the number of clusters is estimated for every split in every permutation. Finally, a cap was obtained by averaging the time frames within each cluster. We first found that there are individual differences in the number of reproducible brain cap states. Across permutations, the occurrence rate of estimating 4 or 5 cap states were about 50%. Surprisingly, when we looked at the co-occurrence of the same number of caps in both splits, for example, the probability that we get 4 caps from split 1 and also 4 caps from split 2 was extremely low. Because each split helps contain a distinct, non-overlapping subset of samples, we hypothesized that individual differences in the number of brain states plays a role in the observed between-split differences. To test this hypothesis, we quantified the individual's preference toward a specific number of caps by the probability of occurrence. In this scatter plot, the data point is a single subject. On x-axis is the number of occurrences of estimating 5 caps out of 1,000 permutations minus the number of occurrences of estimating 4 caps when this specific subject is involved in split 1. On y-axis is the same measure estimated from split 2 data. As you can see, there's a very clear correspondence between two splits, suggesting the presence of a cap state that is reproducibly found in a subset of subjects, but not in others. Using a hierarchical clustering of these caps across 1,000 permutations, we identified the basis caps. Spatial correlation between the basis caps from either 4 or 5 cap sets revealed two pairs of caps with anti-correlation. In addition to cap 3, which was only found in the 5 cap basis set, but not in the 4 cap basis set. When compared to predefined resting state networks, cap 1 involved a strong bipolarity between the default mode and vertebral networks versus the dorsal attention, cinguloopercular, somatomotor, and secondary visual networks. CAP2 exhibited a weaker bipolarity between them, but also included the primary visual and orbital affective networks. CAP3 showed a strong bipolarity between a different set of networks. Next, We wanted to evaluate if spatial topography of the estimated caps are reproducible across permutations. We computed the spatial correlation between the estimated caps in each split to the basis cap set of template caps and collected the correlation over permutation to draw a marginal distribution of correlation values. We confirmed that whenever four caps are estimated from a split data, their spatial patterns are reproducible. The same for the cases of five cap solutions. Importantly, when four caps are estimated from a split, cap 3 was reproducibly missing when compared to the five cap solutions. Next, we evaluated if temporal properties of the caps are reproducible across permutations. The color bar indicates the caps estimated for every time frame in a group concatenated time series data from a single split. Different color indicates different cap states. So for a blue state, we can measure three neural metrics. The first is fractional occupancy, the total number of time frames that are identified as a blue cap, in addition to the within subject mean and standard deviation of dwell times, which are measured from each continuous time segment. We measure those for day 1 and 2 separately. Indeed, these neural measures were reproducible at the single subject level across permutations. Here, subjects were rank ordered by the within subject mean of dwell time on CAP1 positive state, estimated from day 1 split 1 data. The measures were reproducible across permutations and across split, as you can see from the colors, but not across the 2 days, suggesting a substantial amount of day-to-day variability. The result was also evident when we compared the between-day reliability at the single subject level more specifically, using the linear regression model on the left side and the intra-class correlation coefficient ICC on the right side. The range of estimated ICC in neural measures were around 0.4, which was similar to, actually slightly above than the average ICC values reported in the recent meta-analysis. We can project the caps based on their trait variability, indexed by neural measures for each day. A data point indicates a cap state in 1 day. This gives us an opportunity to perform a joint analysis of state and trait neural variations, where state in this plot refers to day-to-day variability. For example, CAP2 has the largest between subject variability in all neural measures than other CAPs, but lower within subject variability. Using this framework, one can pinpoint a brain state of interest that can answer to specific research questions. An important and interesting question would be then whether neural features with distinct patterns of state trait variation can provide information about individual differences. In other words, is there a low-dimensional geometry of neural state trait variation capturing individual differences, which can in turn be related to behavioral phenotypes? We first collected 30 neural features estimated for each individual, 3 neural measures times 5 caps times 2 days. We performed the agglomerative hierarchical clustering and found 3 subgroups. The 3 subgroups were distributed in the low-dimensional space represented by the first 3 neural PCs, which together explain about 73% of variance. We found that individuals exhibiting high scores on neural PC1 occupy CAPS 1 and 3 for a relatively longer time, whereas individuals with low PC1 scores occupy CAP2 state for a longer time. The NeuroPC2 highlights a general pattern of state persistence. That means high within subject mean and standard deviation of dwell time, indicating that when a subject enters to a brain state, then spend a longer time before transitioning to another state. NeuroPC3 captures a specific component of day-to-day variability. The CAP-specific patterns observed in EuroPC1 can undergo systematic changes between days, like sign flip patterns. This, in turn, suggests that patterns of within subset variations, state features, can also be uniquely individualized, just like trait features. We also applied PCA on 262 behavior measures to estimate low-dimensional geometries across multiple behavioral domains. Using a permutation testing, we found 27 PUCs that accounted for a proportion of variance that exceeded chance and considered the first 15 PUCs that explained 50% of the total variance. Reproducibility of these PCs was also evaluated using a split half permutation. The behavioral PC1 highlighted individual life function outcomes associated with cognitive function, emotion regulation, and alcohol and substance use. The variables of working memory task performances have the highest loadings on the behavioral PC1, followed by the emotion, relational, languages, gambling task performances, fluid intelligence, self-regulation and impulsivity, and episodic memory. In contrast, variables associated with alcohol and substance use and psychiatric dimensions showed the lowest negative loadings. Next, we were interested in studying to what extent individual variability quantified on this low-dimensional neurofeature space was linked to variations of individual human behavior. On behavioral PC1, individual scores were different between subgroup 1 and 2 who were classified using the neuromeasures. Individual scores on the behavioral PC1 are associated with the neuro PC1 scores as well as sex. This neurobehavioral association was highly reproducible across permutation and strongly significant when compared to null data. To remind, high NeuroPC1 scores indicates longer occupancy and dwell time in the CAP state 1 and 3 and shorter occupancy in the CAP state 2. We repeated the same analysis for predicting behavioral PC2 that highlights emotion, personality, and psychiatric life functions. NeuroPC2 showed reproducible association with behavioral PC2, whereas age and sex showed larger predictive power. To remind, high NeuroPC2 scores indicate longer persistence, which means longer dwell time in all CAP states. On the other hand, NeuroPC2 and 3 were predictive of behavior PC3, highlighting a strong association with alcohol consumption habits and sex, as well as day-to-day variation reflected in NeuroPC3. I also want to remind you that some people have stronger preference to occupy CAP3 than other people. Finally, we studied whether and how the presence of CAP3 is related to individual behavior. As expected, subgroup C had a high probability of having CAP3. Subjects with a high probability of CAP3 occurrence showed low scores on NeuroPC1 and high scores on NeuroPC2. Indeed, we found a weak negative correlation between the probability of CAP3 occurrence and individual scores of behavioral PC1. We found no correlation between the probability of CAP3 occurrence and behavioral PC2 and 3. In summary, individuals with high scores on behavioral PC1 who have higher cognition and emotional regulation and low nicotine consumption habits showed these characteristics. They spend longer time at CAP1 than at CAP2 state, have higher between subject variance and lower within subject variance at CAP1 than at CAP2, show high global persistence, longer dwell time in all CAPs, have lower chance to have CAP3, and were associated with sex differences. High scores on behavioral PC2 show longer dwell time in all CAPs, spent longer time in CAP1 than in CAP2, and were associated with age. High scores on behavioral PC3 was associated with longer dwell time in all CAPs and a higher chance to occupy CAP3 state, and also being associated with sex differences. In conclusion, we found that human brain manifests a highly reproducible, low-dimensional set of features that index brain-wide co-activation patterns detected using neural imaging resting state fMRI. The neural feature reduction captures a general motif of individual variation, such that individuals occupy this low-dimensional state trait neural space differently, which in turn predicts life and behavioral outcomes. State variance indexed by day-to-day variability in co-activation dynamics are loaded on this low-dimensional motif of individual variation, therefore reflecting neurophenotypes. Principal variations of neural state and trait cap features co-vary with the principal variations of behavioral phenotypes. Finally, person-specific probability of occupying a given cap was highly reproducible and associated with the neural and behavioral features. This work had several limitations. We did not test the impact of sample size and the amount of data. We did not test if these findings are generalizable to a larger and more diverse samples of time scales, like over days, months, or years. Future studies that takes into account heritability and in clinical and developmental populations will be of great interest to understand heterogeneity and state and trait neuroprofiles associated with neuropsychiatric or neurological symptoms. Thank you for your attention to this talk.